it's about time we are start. So first of all, let's welcome all the judges. It's a great pleasure to see everybody here, and this is our first English speak competition initiated by our dean, Professor Lian Zhenzhang. And actually, I think some of you might know that initially this event should be in May, right? And due to the pandemic issue, now we finally can make it happen. So first of all, let's welcome our dean to give us a little speech.
Former Vice President Gao Langxian and this Chief Operating Officer, Professor Zheng or Dr. Zheng, and distinguished guests and colleagues, and also ladies and gentlemen. So good afternoon. And first of all, I want to welcome you to this important event. And next, I want to say a few thanks to people who help organizing this committee. So first, thanks to Dr. Zhang Peijing, Amy, and the staff members, Peijing and Yafu, and Melody, Xianlu, and who else? I hope I don't miss someone. Maybe Huang Linlin, for helping organizing this committee event, organizing this great event. And also, thanks to Yifu Shenjun for long-term support and their financial support to make this event possible. And finally, I also want to thank our committee members for serving on this evaluation committee, and in addition to Professor Gao Langxian and Dr. Zheng Yin Yunzang. Okay, so as you know, our college CLS stands for College of Life Sciences. Of course, someone may say LS also stands for Langxian, right? But I also have another interpretation of this College of Life Sciences. LS means leading scientists, and C, after the merge of a university, I interpret the C is the cultivating. So cultivating leading scientists as an educator is a very important mission for us. So what is the important ingredient of the leading scientist? I think is the spoken and written communications. And that's the reason today we have this event. And I think spoken communication is particularly important and is the key for interdisciplinary collaborations. So I often told my students that you need to present your data or your work to your grandma or your grandpa, and to make them understand. If they can understand, that means your presentation is very successful. And in addition to clear presentation, I think timing or time matters. Time is also very important. So as a scientist, we need to be able to summarize or to present your work or tell your story to people outside your field within a controlled time or a very short time, such as 1 or 3 minutes. And that is so-called an elevator speech, right? So I think that's a very important skill for leading scientists. So today, I think we have this event, and I think this is just the first step toward promoting our students' scientific presentations. And I hope you can enjoy today's mental food. I think this is a kind of mental food. If you don't have your lunch, just enjoy this mental food. And after you finish your presentation, I think we also prepare a lunch box for students, right? So they can also enjoy their meal outside. So finally, I also wish that this event successful Thank you, and of course, thanks again for Amy for organizing this event. Thank you.
Thank you again for our team to initiate this great activity. And I think all of you are very lucky because we have a great judge today to help you score your presentation. And the first one is Dean already mentioned that actually she is our previous dean and our previous vice president, also my teacher, Professor Lang Xiangao. And we'd like to thank the CEO of Binet Biomedicine, Dr. Wan Changzhen. And I think that the next judge is the director of Institute of Neuroscience, Professor Yawei Zheng. And our director, IMI, Nian Rong Chen. And another great teacher in Institute of Neuroscience, Professor Shijie Lin. And the professor from Institute of Physiology of Mind and Cognition, Professor Li Hongzhang. And the professor from Department of Life Science and the Institute of Genome Science, Qingwei Luo. And Weiqian Yuan. And the professor from Institute of Biochemistry and Molecular Biology, the institute I graduated from, is Professor Jiarong Huang. And another two professors from our institute is Minghan Cai and Yuhuan Cai. And the professor from Institute of Biomedical Informatics, Zhen Qinglin. Okay, so this is all our judges today. And before we start, I'd like to briefly summarize the rule today. First of all, when you go up there, you hold the microphone. We start counting down. And in 2 minutes and 30 seconds, you will see the plate raising. And 30 minutes is time up. We don't want to scare you, but the time, as Dean mentioned, the time control is really important. So we have to stop you at 3 minutes exactly. And don't bring anything there because we prepare everything there for you. Or maybe we can start it.
In my talk today, I'll briefly describe some of our highlights of our study about the neural substrate for animals' innate behaviors.
Basal forebrain is a complex nuclei located in the ventral part of the forebrain area, containing different types of neurons. Our lab is interested in
cells are identified to play a role in neutrophilic asthma. For example, when bacterial infection, air pollution stimulate lung mucosa.
Hello everyone, I'm Alice, and my topic is focused on a novel protein JHP0417 and try to investigate its role in
heavier levels of these mice. The neurological function in sensory motor getting is impaired in patients with schizophrenia.
EOA and sucrose gradient experiment to identify dynein FOXP2 knockout cells. The overexpression of dynein 1 could lead to the collapse
where the plasma membrane will eventually rupture and release the net component. So first, we will
the nucleoside transporter that located on the membrane of lysosome and mitochondria.
In animals' mouth and in animals' brain, the adhesion molecule is very important for cell formation, and citracone is one of them.
In cell initiation, there are two critical proteins C1C4 will recruit TPK2 on the centrosome and promote cell formation.
Imagine yourself swimming in beers and wines or up to 15% of alcohol for about a month.
Double kills can predict reward in training stage. In every 2 seconds, one of the like kill and one of the cell kill will present simultaneously. So there's
whether or not you have inherited epigenetic marks from your parents, grandparents, or even great-grandparents. Maybe you do or maybe you don't.
Focus on the amygdala to the dorsal striatum. And first, we inject two retrograde tracer into the dorsal and the ventral.
Degenerative disease with a symptom of memory loss. Even though every 3 seconds someone will develop dementia, there's still no effective
elevated increased level in patient of the chronic migraine. And throughout the whole branch, we found pinpointed specifically in the central region of
protein with a conserved carbohydrate recognition domain CRD. And Glytin 3 is the only chimeric type in Glytin family. With what
sounds great when we are talking about the food, right? But when we are discussing about our body composition, it is not happy to see too much fat accumulate.
Can rarely infect epithelial cells, so it makes it impossible and very difficult to study EBV infections in epithelial cells.
Typical treat to ask for sweet, but not only does the children love sweet, so does adult like me. Yeah, we drink bubble milk tea.
90% of cases with mutation or overactivation. Therefore, afatinib, this is an EGFR inhibitor, is expected to be an effective treatment
in the lymphoid cell, ILC2. So what is ILC2? There are 19 non-being lymphocytes, and which can be activated by our
regulum panel. Here we use in the human whole blood sample. And indeed, our RS regulum is enriched in different levels in the immune cell types.
To the LHB, we hypothesize that they may be a part of the aversion system and may oppose the reward processing through the LHB.
UPT5. They form a complex named DSIF and interact with RNA polymerase II.
Is in the immunocompromised patients, so that's why we target on this disease. Okay, and the uropathogenesis is
Rule prediction error, or RPE, is the discrepancy between actual and predicted reward value, which has long
not simple as those we handle in vitro. Moreover, because the adhesion signal will have spatial separation,